Okay. So 4 stages of glucose metabolism. Okay, stage 1 is we break down large molecules to small. That means proteins to amino acids, carbohydrates to glucose, and then fats to fatty acids. Now, the fatty acids, if we run out of glucose molecules, the fatty acids can be converted to glucose, and so can amino acids. Both of those all feed into the glucose. Now, second stage is glycolysis. And glycolysis takes glucose to pyruvate and acetyl-CoA. And we produce 2 adenosine triphosphates. Now, if you're a yeast, that's it. You only get out of 1 molecule of glucose, you get 2 ATP, as opposed to us, which gets 38 ATP. Okay. Then the Krebs cycle, its main benefit is it produces the NADH and the FADH2 coenzymes. It also produces 2 ATP. Because now the coenzymes, the FAD2 and the NADH, feed into electron transport. In the electron transport, there are 2 parts to the electron transport. They usually don't mention the second part, which is called the phosphorylation process, which takes and puts a phosphate group on adenosine diphosphate. So 2 ATP from glycolysis, 2 ATP from Krebs, and 34 from electron transport, giving us a total of 38. Oops. Now we're just from now on, we're just talking about electron transport. For every NADH oxidized, we produce 3 ATP. For every FADH2, we produce 2 ATP. And this is what it looks like. The NADH, when it's oxidized, it goes to NAD plus, H plus, and 2 electrons. Something similar to FAD, it produces FAD 2 hydrogen ions and 2 electrons. Now, I'm going to talk about the mitochondria in a minute here. But basically, we go through a series of steps now. And the word electron transport means we're moving electrons from one protein to another, all in the mitochondria. There are a series of steps. There are 4 steps. And then eventually we get down to this guy here at the bottom of the series, where we actually take oxygen, and this is the reason why we need to breathe. We take oxygen and reduce it to water. So oxygen gets 2, for each half mole of oxygen, it gets 2 moles of hydrogen atoms, hence the water, H2O. Of course, you can't have fractions, coefficients, so if we get rid of the fractions, 4 electrons, 4 H pluses, 1 oxygen molecule gives us 2 waters. Now, going back to the NADH being oxidized. The first place it gets oxidized, something called CoQ. Now, if you go to CVS or you go to Costco and you go into their pharmacy area, you'll see all kinds of CoQ supplements. And the reason why they're offering it is because in case you have a deficit of CoQ, you cannot metabolize glucose. So they want you to always have that. Cytochrome C is another one. And each time the electron's transferred, there's some energy being given off each time. So we go from, and there's a number of cytochromes, but we start oxidizing the NADH and the FADH2. First, it enters the CoQ and gives off energy. And we use that energy to move the electrons and hydrogen ions across the inner membrane of the mitochondria. I'm going to show you that in a minute. And then they move to the cytochrome C. And then we end up with reducing oxygen to water. Okay. All these, now see, we haven't made any ATP yet. All we've done is we've moved across the inner membrane of the mitochondria. So if we take a crude picture of mitochondria, this is the outside of the mitochondria, and all energy is made there, assuming it's a eukaryotic cell, not a prokaryotic cell, meaning it has a nucleus. Okay, so if we look at this thing, this is the outer membrane here, and this is the inner membrane here. So if we take a snapshot of that and made it bigger, and this slide here is under slides on Canvas, so you don't have to draw that. Okay, so we have these big protein complexes here, 1 through 4. And the purpose, what they do is they take the H plus and bring it up through there. One through 4. Once it's there, you end up with this gradient of too much hydrogen plus there. So it wants to come back across. The only place it can come back across is over here in protein complex 5. Now, this is the matrix here. This is that squiggly line here, that membrane. Oops. This here. So we're talking about matrix. This here is the intermitochondrial membrane. And then this here is the intermembrane space right here. But this is where all the action is. If you look at that under a microscope. Okay. So with the enzyme ATP synthase, it converts the H pluses as they want to come back down to the matrix. And when they come back down through the matrix, what happens is ADP plus phosphate becomes ATP. So it's a circular process. So the first 4 proteins, the CoQs, the cytochromes, etc., move the H pluses across the barrier, the membrane. And then they start building up big time charge there, charge imbalance. So through osmosis, those H pluses want to come back, but they can't come back the way they came. The only way they can come back is through the ATP synthase protein or enzyme. As they come back down, that's when they produce the ATP. Okay, so these protein complexes, their purpose is to move H pluses from the matrix to the intermembrane. That's their purpose. The ATP synthase complex purpose is to move the H's back to the matrix. So they go up and then back down. But there's a selective pathway to go up there. That's through the 4 protein complexes, the CoQs, the cytochromes, et cetera. And there's only one way down, and that's through the ATP synthase. So every time one comes down, these two get added together to form adenosine triphosphate. Now, repeating what I said earlier, each NAD plus creates 3 ATP. And through the various processes, the Krebs cycle and the glycolysis, we've created 10 NADHs. 10 times 3 is 30. That's the extent of the math in this class. Each FADH2, there's 2 of them created. There are 4 from that one. So the total is 34. Now this is that's the electron transport, okay? And we get 2 from glycolysis, 2 from Krebs cycle, gives us 38 ATP per glucose. As opposed to a yeast that gets 2. Now, this is a very, very simplified version of this. It gets incredibly complex, but I wanted to simplify it. For those of you who have a book, the book is much more complicated than this. And frankly, I think it's so complicated, it gets very confusing. So I took the kind of the bird's eye view of all this. Okay, now your test next week is going to be open notes, okay, or unit 5. And I'll put together a summary of what I want you to make sure you understand. Secondly, it's going to be semi-comprehensive. So there's going to be a section from earlier units, a few reactions, a little bit of naming. And you're going to have 2 hours in here. And we start at 12:45 on Tuesday. Okay, so that's it for the semester, content-wise. Thursday, I'd like to do some review. Also, I'm going to have some questions too, study questions. No, I hate that. When last week you asked me when the tests are going to be graded, and I said, "Oh yeah, a week later." No, they're going to be. Hopefully, I get them up tonight. That's the goal. Of course, I have lofty goals sometimes, but that's the goal. Okay, any other questions? I know see a number of you have posters. Now, for the people that already looked at your posters, make sure you send me an email with a picture of your poster plus your essay. Okay? Now, a week from today is going to be your last opportunity to submit a poster and essay. So you still have time. Okay? Yeah. Yeah, like your emails keep asking me. And thank you for doing that. Keeps me. Okay, so any other questions? Okay, cool.